Okay, so we know ATP is important and that it is one of the most important molecules carrying cellular energy that fuels metabolic reactions. So obviously it's really important. We talked about the 3 ways to get it. And now what I want to talk about is 2 of the major energy generating pathways that exist. So we're going to start with cellular respiration and some modifications that kind of come off of cellular respiration. Fermentation being one of them. And then in the next lecture, you'll hear about photosynthesis. All right, so let's talk about cellular respiration. All right, so first off, I just want to kind of mention that just like we talked about, everything is sort of linked. Anabolic and catabolic reactions are linked. Consumers and producers in an ecosystem are linked. These major energy generating pathways that we're going to discuss are intimately linked. So the major energy generating pathways we're going to talk about are photosynthesis and cellular respiration. So these, again, when we talk about generating, we're talking about transforming energy, but these are categorized as major energy generating. The type of energy that is generated via photosynthesis is stored energy in the form of chemical bonds. So the process of photosynthesis uses energy from the sun, photons, to release electrons for chemical bonds stored in water. And those electrons are utilized to form bonds between carbons to generate sugar. So it is overall an endergonic process because it's using energy from the sun to create very high power chemical bonds to create large molecules, organic molecules. This is paired with an overall exergonic or catabolic reaction of cellular respiration, where the bonds in those organic molecules generated in photosynthesis are broken. The electrons are released from those bonds, harnessed, and used to generate ATP. So what you're doing in that case is you're generating sort of usable cellular energy. So overall cellular respiration is categorized as an exergonic process. It's kind of releasing a lot of energy and creating or putting it in a form of more usable cellular energy. These are linked because they're products. The products of one are the reactants of another, as we will talk about. So they are intimately linked in the ecosystem. So we're going to start talking about cellular respiration, but before we get there, I want to talk about some commonalities between these 2 major metabolic pathways. And understanding these commonalities will really help us understand these products or these reactions individually as well. So when it comes to cellular respiration and photosynthesis, both of these involve redox reactions. So we'll talk about those. But we talked a little bit about redox reactions in the previous lecture. And remember that with an oxidation reaction, electrons are released, and you have to have some other molecule to basically like grab those released electrons. So in this case, you have what are called electron carriers. So in terms of cellular respiration, you have 2 primary electron carriers. The first is nicotinamide adenine dinucleotide, or let's just call it NAD+. When NAD+ gains an electron, so it's reduced, it also gains a hydrogen, remember? So what happens is NAD+ gains an electron and a hydrogen, and it becomes NADH. Okay, so NAD+ gains an electron to become NADH. So NADH is the reduced form. You see NADH, that H means it's carrying an electron. So NAD is an electron carrier. When it gains an electron, it gains a hydrogen as well. It's called an NADH. The other electron carrier is called flavin adenine dinucleotide, or FAD. When it gains electron, it gains a couple of hydrogens, and it becomes FADH2. So again, you see that hydrogen added, that means it's carrying an electron. Remember that both of those, NADH and FADH2, when they have that hydrogen, that means they've gained an electron. That electron came from an oxidation reaction. All right. So NAD plus and FAD are both used for cellular respiration. Photosynthesis uses another electron carrier that cellular respiration does not, and that is nicotinamide adenine dinucleotide phosphate, or NADP. When it gains an electron and a hydrogen, it becomes NADPH. So again, you can see that when these molecules gain an electron and they are reduced, they also, you can tell because they have that hydrogen as well. So those are the electron carriers. Importantly, again, both of these involve oxidation reactions. They also both have these electron carriers carrying their electrons to an electron transport chain to generate a proton motive force so that we can get a ton of potential energy to generate ATP. So they both utilize that chemiosmosis, electron transport chains, and they both utilize those electron transport chains to generate enough energy to convert ADP into ATP, our very good source of cellular energy. All right, so let's start with aerobic cellular respiration. So another term for aerobic cellular respiration is carbohydrate catabolism, because what we're doing is we're going to take a carbohydrate, sugar, we're going to oxidize it, we're going to break all the bonds in this sugar, release electrons from that sugar, generate a proton motive force, and generate a ton of ATP. The overall reaction for cellular respiration, you can see here, we are using an organic compound. In this case, we're talking about glucose. So you see C6H12O6, which is the chemical formula for glucose. This is a sugar ring. Cellular respiration, it's called aerobic cellular respiration, or at least the one we typically think of is aerobic cellular respiration, because as you can see here, for every carbon, you also need an oxygen. So notice there are 6 carbons in glucose. There's also 6 oxygens that are going into this. And then this goes through the entire process of cellular respiration, which we are going to break into 4 steps. And the products of this reaction are 6 carbon dioxides. Notice I said that you will start with 6 carbons and 6 oxygens and generate 6 carbon dioxides. And then you also release 6 water molecules. And look at all that ATP you get. Oh my God, that's a lot of ATP. Let's talk about it. The overall goal of cellular respiration is to break bonds in carbohydrates to release electrons. So basically oxidize your carbohydrate. You want to harvest those electrons with our electron carriers. In the case of cellular respiration, that's going to be NAD plus and FAD. Those electron carriers are going to carry the electrons to a membrane where they're going to use an electron transport chain to generate a proton motive force. And that proton motive force will be utilized to generate enough energy to add that high energy bond between ADP and a phosphate to generate ATP. So importantly, this occurs in 2 major compartments in a cell. Glycolysis, which is the very first step, occurs in the cytoplasm, but the vast majority of this actually occurs in the mitochondria. So you may have heard that the mitochondria is the powerhouse of the cell. That's because the last 3 steps of cellular respiration occur there. And that's where you get a lot of your ATP. But I just want to remind you that you know what a proton motive force is at this point, and you know that the mitochondria, if you recall from our talk about cell bio, that the mitochondria is made of a membrane with a bunch of folded membrane inside of it. And now you know that the electron transport chain requires a membrane. So the reason that the mitochondria is the powerhouse of the cell is because it's made of membrane. So technically, membrane is the powerhouse of the cell, because all we need to generate an electron transport chain and a proton motive force is membrane. Okay, so glycolysis can occur in the cytoplasm, no big deal. But the rest of these steps, the electron transport chain, requires a membrane. So takes place in the mitochondria, in eukaryotes. All right. So this is just kind of a summary of the steps. You'll see a lot of summaries for cellular respiration throughout this presentation, because I'm all about summaries. For the steps of cellular respiration, again, we're going to break it into 4 steps. We're going to break it into step 1, which is called glycolysis. Just want to give this a breakdown of that term. Glyc is referring to the fact that we are starting with a sugar, glucose. But the prefix glyc tells you it's a sugar. And lysis means to break something apart. So if you see the term glycolysis, you should infer from that that you're breaking apart a sugar. The first step of this is we're just going to break a sugar in half. All right, glycolysis. Step 2 is called the intermediate stage, where we break some more bonds. Step 3 is the Krebs cycle, where we break some more bonds. The first 3 steps of cellular respiration collectively serve to completely oxidize glucose. And remember, whenever we say oxidize, that means you're breaking bonds and releasing electrons. So the first 3 steps of cellular respiration serve to break down glucose, break down that sugar, to release as many electrons as possible. And then, with those electrons, you enter the 4th step, which is the electron transport chain. So then all of the electrons you released via the first 3 steps are used in an electron transport chain to generate a proton motive force and generate a ton of ATP. All right. So you will notice that in this picture, over here to the right, you have the first 3 steps, glycolysis being the first, intermediate being the second, shown in gray, Krebs cycle shown in green. This is the first 3 steps, releasing a ton of electrons. Remember that NAD is our electron carrier, FAD is an electron carrier. We're generating NADH, we're generating FADH2, we're generating NADH. That means we have a bunch of electrons being carried to the electron transport chain, again, to generate a proton motive force, to generate ATP. So something I want to point out here that we'll talk about later is that obviously we are working to store those energies, those electrons, in the bond that is ATP. But then we don't want to just get rid of the hydrogens that we used and some extra electrons that we've got hanging out. So what we have at the end of the electron transport chain is we have a molecule that serves to kind of like clean everything up. And in the case of aerobic cellular respiration, that molecule that serves as sort of just like the final electron acceptor is going to be oxygen, which is why this is aerobic cellular respiration. Oxygen is our what we call our final electron acceptor. So it binds up all of the electrons that we used in the electron transport chain after we use them to create ATP. And as we will find out, that generates water. All right. So that's a general summary. let's get into it. So this is just looking again at where each of these steps take place. As I mentioned, the very first step of cellular respiration, which is glycolysis, which is where we take glucose and split it in half into glucose is a 6-carbon molecule. We split it in half. That means we get 2 3-carbon molecules. Those 3-carbon molecules are called pyruvate. So what happens, that glycolysis, the splitting of 6-carbon glucose into 3-carbon pyruvate, occurs in the cytosol. This does give us a little ATP, as we'll find out, via substrate level phosphorylation. Then that pyruvate enters the mitochondria, and we undergo the intermediate stage, as well as the Kreb cycle, which is also referred to as the citric acid cycle. We get a little more ATP via substrate level phosphorylation, but primarily we are getting the release of electrons, and those electrons are being carried by our electron carriers all the way, still in the mitochondria, to a membrane, where we generate a proton motive force via an electron transport chain, which gives us a ton of ATP. So you can see here, this is very important, we have these 4 steps. And you can see that in 2 of these steps, you get some, notice that the BAM is smaller, you get a little bit of ATP. You get a little bit of ATP via glycolysis. You get a little bit of ATP during the Krebs cycle. But notice that overall, you get way more via oxidative phosphorylation through that electron transport chain. So we'll talk about that. All right, so let's go through these 4 steps. Again, 4 steps. The first 3 are working to fully oxidize glucose, release all of the electrons from glucose, and then the 4th step is just going to be using those electrons to generate ATP. So our first step is glycolysis. Again, breaking sugar. So what we're starting with in our metabolism in cellular respiration is a sugar. In this case, we're starting with glucose. Glucose is composed of 6 carbons. Remember, its structure is C6H12O6. All we're concerned about with cellular respiration is the carbons in that molecule. So you can see here to the right, I drew glucose. Glucose has 1, 2, 3, 4, 5, 6 carbons attached by bonds. So that's a 6C is referring to the fact that this molecule has 6 carbons, 6-carbon glucose. And what we need to do through glycolysis, again, lysis meaning split it, what we want to do is break glucose in half. Basically just go and separate that 6-carbon molecule into 2 3-carbon molecules. And each of those 3-carbon molecules are called pyruvate. So in order to break the 6-carbon ring into 2 3-carbon molecules, we have to break 2 bonds. So what I want you to do now, and to do these calculations, is every time you notice we break a bond, think about that bond breaking, releasing 1 electron. And every time you release 1 electron, right, you have to have something to receive that electron, because we want to carry those electrons to the electron transport chain. So the reason I say that is because here what we're doing is we're taking glucose, we're breaking a bond here, we're breaking a bond here. Breaking this bond, we release an electron, and we need an NAD plus to grab that electron to be reduced, and therefore we generate an NAD. We're breaking a bond here, therefore releasing an electron, we need an NAD plus, and therefore we generate an NADH. So what that means is, notice again, in 6-carbon glucose, we're breaking 2 bonds. We're releasing 2 electrons. Therefore, we need 2 NAD plus. And if we had 2 NAD plus, grabbing 2 electrons, 1 from each of the bonds, we're going to end up with 2 NADH. Okay, so again, what we're doing here is we're breaking glucose in half. In order to do that, we're breaking 2 bonds. Each of those bonds release an electron. You need an NAD plus for each of those electrons. So overall, breaking glucose in half would give you 2 NADH. You have 2 electrons, 2 NADH. Okay. So another thing is getting this all started kind of needs like a kickstart. Okay. So this is the only step of cellular respiration where we kind of need this kickstart. Like, let's get this started. So glycolysis actually does require a little bit of energy investment. And this energy investment requires 2 ATP. So you can see here up top in our reactants, we have 6-carbon glucose, and you'll see plus 2 ATP. 2 ATP kickstart this entire process. Okay. Another thing that happens during glycolysis is that when these bonds are broken, we're not going to go into the individual steps here, but when these bonds are broken, a couple other things happen. What happens is you actually get the release of some phosphates and a lot of energy. You get enough energy released right there and phosphates released right there to generate a little bit of ATP. Because it's occurring right there and there's no electron transport chain, that little bit of ATP that's generated right there, you see right here in the products, you have 4 ATP, that's generated via substrate level phosphorylation. Okay, so overall process of glycolysis, what we're having, sorry, my cat is snoring so loud right here. Yeah, you. All right. What we have is we have a 6-carbon sugar. We break that sugar in half into 2 3-carbon molecules. In order to do that, we had to break 2 bonds. From each of those bonds, we released an electron. An electron from here bound to NAD plus to give us 1 NADH. An electron from this bond breaking reduced NAD plus to give us our second NADH. So we get 2 NADH. That breaking of those 2 bonds is so powerful that each of those bonds was able to fuel the production of 2 ATP via substrate level phosphorylation. So notice that at no point during this reaction was oxygen involved at all. So glycolysis is also characterized as, in cellular respiration, the only stage of aerobic cellular respiration that does not require oxygen. And oxygen is at no point involved in this process. So glycolysis is an anaerobic process. That doesn't mean it's anaerobic cellular respiration, which we'll talk about. It just means that oxygen is not involved. Okay, so we broke glucose in half. We used 2 ATP to kickstart it. We ended up generating 4 ATP via substrate level phosphorylation. We released 2 electrons, and therefore we were able to generate 2 NADH. And the organic product of this reaction, products of this reaction are 2 3-carbon molecules. So we went from 1 6-carbon sugar to 2 3-carbon pyruvates. Okay, so what we need to do is keep track of the energy we have created so far. So you can see my energy tally over at the top right. You can see that via this process, I have those 2 NADH recorded. So those are carrying our energy in the form of electrons. You'll notice that in the energy tally, I have 2 ATP. That's because even though we were able to generate 4 ATP via this process, we had to invest or use 2 to kickstart it. So whenever we talk about our energy through this process, what we want to calculate is how much usable energy we've generated. Even though we generated 4 ATP in this process, because we had to use 2, we really only gained 2 ATP, because like using 2 kind of neutralizes 2 of the ATP we get out of this process. So in our energy tally, we say we only generated 2 ATP. The reason we say 2 ATP is because that's a net increase in only 2, because we ended up having to invest 2. Okay, so let's go through this again. We start with 6 carbon glucose. We break 2 bonds to split glucose in half. My fingers are disappearing. We break 2 bonds. We start with 6 carbon glucose and we break 2 bonds in that 6 carbon molecule. 1 here, 1 here. That splits that 6 carbon molecule into 2 3 carbon pyruvates. When we break those bonds, it's oxidized. Electrons are released from each of those bonds. The electron here is bound up to NAD+ to make an NADH. The electron released from this bond is bound up to NAD+ to make our second NADH. 2 ATP are used, 4 ATP are generated via substrate level phosphorylation. Because we utilized 2, even though we gained 4, we kind of subtract the 2 that we invested. So that means we have a net gain of 2 ATP. We also have 2 electrons. So from this process, we used 2 ATP, we used glucose, those are our reactants, and our products are 2 3 carbon molecules, pyruvates, 4 ATP, 2 NADH, but again, our energy, our overall energy gain is only 2 ATP. Substrate level phosphorylation. How do I know that this is substrate level phosphorylation used to form those ATP? All you have to ask yourself is, did we talk about an electron transport chain yet? No. If we get ATP and you don't notice an electron transport chain involved in it, it's substrate level phosphorylation. Okay, so again, we have some energy generated through this process, net 2 ATP. You'll also have some NADH. But what we're going to track now is we have those 2 3 carbon pyruvates. So what are we going to do with those? Well, those enter the intermediate stage. So with the intermediate stage, all we're doing is we are going to continue to break down that organic molecule. We're also in the intermediate stage going to kind of like prepare ourselves to enter the Kreb cycle. The Kreb cycle is a pretty big process. So we're going to give ourselves kind of a leg up for that process during the intermediate stage. This stage is sometimes also referred to as like acetyl-CoA production. I just like calling it the intermediate stage because it makes sense. Okay, so what we're starting with in the intermediate stage is the product of glycolysis, which is a 3 carbon pyruvate. And what we're going to do is we're going to break a bond in that 3 carbon pyruvate. This releases a carbon. So we don't really want just like carbon hanging out. We want to be able to recycle it. So we can actually recycle carbon by attaching an oxygen to that carbon, and then we can just like breathe it out. And if we breathe it out, it can be used for other things like photosynthesis to make more sugar. So one of the first things that happens is with the intermediate stage, we take the product of glycolysis, which was a 3 carbon pyruvate. We break that bond between 2 of the carbons in that 3 carbon molecule. This releases a carbon. You'll notice that this is the first time that we require oxygen. So the intermediate stage does require oxygen. Anytime you see a carbon released, right here, we're having a carbon released, you need an oxygen to bind it up. So this requires oxygen because you're releasing a carbon. And in this stage, you generate carbon dioxide because that oxygen binds up that free carbon and carries it out of the body. So we're breaking a bond, releasing a carbon, binding it up with oxygen to create carbon dioxide. Also, what happens when we break a bond, remember that's releasing an electron, so pyruvate is oxidized, and that electron is released. We use NAD+ to bind it up, and therefore you'll see we break a bond, we get an NADH. Another thing that we do, because this is the stage before we enter the Krebs cycle, and the Krebs cycle is a pretty big metabolic pathway, as we will see, is we want to give ourselves a leg up. So remember when we talked about enzyme function in the last lecture, we said that coenzymes are organic molecules that can kind of help speed up chemical reactions or help speed up enzymes. So one thing that we do during the intermediate stage is we add a coenzyme to our resultant 2-carbon molecule. So whenever we chopped off this carbon, broke that bond, released the electron, generated NADH, what we do with the resulting 2-carbon compound is we add a coenzyme. So this becomes what we call acetyl-CoA, or acetyl coenzyme A. So this 2-carbon molecule is called acetyl, and then it's attached to coenzyme A. Okay. So what happens here, again, is we start with a 3-carbon molecule. We break a bond, which releases an electron. That's where we get NADH. It also releases a carbon. This attaches to oxygen, which gives us carbon dioxide. And then we use this coenzyme to attach to our resultant 2-carbon molecule to generate acetyl-CoA. So in this picture here, you are looking at what happens to 1 pyruvate. With every 1 pyruvate, you're going to get 1 carbon dioxide and 1 NADH. But remember, through glycolysis, we actually generated 2 pyruvate, right? We split 6-carbon glucose into 2 3-carbon molecules. So we have actually 2 pyruvates. And the same thing will happen to both of those. So what that means is you're looking at what happens to 1 pyruvate. So realistically, if we were looking at all of cellular respiration, this is going to happen to both of those, which means that you would actually get 2 carbon dioxide, 1 for each of the pyruvates. And you would also get 2 NADH, 1 for each of the bonds broken. So that's why you see with our energy tally up at the top, you see, again, during glycolysis, we got the 2 ATP via substrate level phosphorylation. You got the 2 NADH when those bonds in glucose were oxidized. During the intermediate stage, because you're oxidizing 1 bond in each pyruvate, right? You have 2 pyruvates, you're breaking a bond in each of them. That will release 2 electrons total, 1 from each of those pyruvates. So you're going to get 2 NADH. That's why you have that in that energy tally. So again, this step is aerobic. It does require oxygen. It requires oxygen because we're breaking off carbon. We get the production of carbon dioxide as a byproduct. So what goes into the intermediate stage is 2 pyruvate, 2 oxygen, 1 for each pyruvate, and 2 coenzymes. And what comes out of this is 2 acetyl-CoAs, 2 carbon dioxides, and 2 NADH, right? Okay, so that is the intermediate stage, or acetyl-coenzyme A production, or pyruvate preparation. It's called a million different things. I call it the intermediate stage. It's sometimes called pyruvate oxidation, because that's technically what you're doing. You're oxidizing pyruvate. Okay, so now we've got acetyl-coenzyme A, and we got to break it down. How do we do that? Well, I hope this isn't too outdated because I loved Game of Thrones, but I miss it so much. But for those of you that get your reference, brace yourself. The Krebs cycle is coming. Winter is certainly not coming because it was like 100 degrees today. But brace yourself, the Krebs cycle is coming. So what do we do with that resultant acetyl-CoA? We move into the Krebs cycle. Okay, so first off, yes, every time I pull this slide up, people are like, oh God. But listen, it is not that crazy. The reason it's not that crazy is because I don't need you to know this cycle. Don't memorize this cycle, okay? I'm going to tell you exactly what you need to know about it. Look at the top here. you'll see that you have our product from pyruvate oxidation, or the intermediate stage, which was acetyl coenzyme A. That is what is entering. Remember, we started with glucose, we split glucose in half into 2 pyruvates, and we broke those pyruvates in half, and now we have 2 acetyl coenzyme A. So what you're looking at is what happens individually to each of those. All right, so some of you may hear it like, oh, the Krebs cycle occurs twice. The Krebs cycle occurs once, but for each of the acetyl coenzyme A's, and you have 2 of those. All right, so we're gonna see what happens to each of those acetyl coenzyme A's. So we have that molecule, it's a 2-carbon molecule attached to a coenzyme. The first thing that happens, so you can see that 2-carbon coenzyme here. The first thing that happens, and this actually requires water, it's called a hydrolysis reaction, is that acetyl coenzyme A is combined with a molecule called oxaloacetate. That is a 4-carbon molecule. But when we attach our 2-carbon acetyl-CoA to oxaloacetate, it becomes a 6-carbon compound called citrate. So that oxaloacetate is a recycled compound. So the reason it's called a cycle is because we use oxaloacetate repeatedly. It's used to receive those acetyl-CoA and generate citrate, which is why this is also called the TCA or the citrate cycle, because it's talking about the cyclic generation of citrate. But anyway, that's beside the point. What happens, you have your 2-carbon compound come in here, join with oxaloacetate to form citrate. Now you have a 6-carbon molecule. All of a sudden, you have way more bonds, right? We were sitting here being like, okay, I have acetyl-CoA, so I have like 1 more bond I can break and release electrons. Never mind. You enter the Krebs cycle, that 2-carbon molecule combines with oxaloacetate, and all of a sudden you have this giant 6-carbon molecule with way more bonds to break. The more bonds you have, the more electrons you have to release, the more NADH you can generate. So again, some more chemical reactions take place. I'm not going to have you remember each of these chemical reactions, but what I want you to pay attention to is a couple different steps. What happens is, through the Krebs cycle, I don't want to tell you to memorize stuff, but at this point, for simplicity's sake, memorize this. What will happen is at one point, again, for each acetyl-CoA that enters, you'll get an electron released here, you'll have some NADH produced, a carbon released. Every time you release carbon, it binds with oxygen to create carbon dioxide. So remember, if you're creating carbon dioxide, that means it required oxygen. So the Krebs cycle does require oxygen. So here you had an oxidation reaction. You're generating some NADH, you're releasing some oxygen, and generating carbon dioxide. So here you have 1 NADH for every acetyl-CoA that enters. Next chemical reaction, you are having another oxidation reaction. You're generating some more NADH. You also have some carbon released, generate carbon dioxide as a byproduct. Next, you can see at the bottom of the cycle that you actually have a substrate level phosphorylation event, and you generate some ATP via substrate level phosphorylation. And then you have another oxidation reaction. In this case, instead of using NAD+, you're using FAD and generating some FADH2. And then a little bit later, you have 1 more oxidation event take place, and therefore you're generating some more NADH. So if we look at this, what happens is that with every 1 acetyl coenzyme A that enters the Krebs cycle, you're generating 1, 2, 3 NADH, you're generating 1 FADH2, and you're also generating 1 ATP via substrate level phosphorylation. It's for every 1 acetyl-CoA that enters. But remember, you have 2 acetyl-CoAs because you start with 6-carbon glucose, generate 2 pyruvate, generate 2 acetyl-CoA, and so you have 2 acetyl-CoA that enter the Krebs cycle. That means if every 1 acetyl-CoA generates 3 NADH, 1 FADH2, and 1 ATP, then that means that this entire process, because we have 2 acetyl coenzyme A's, we're going to end up with 6 NADH, 2 FADH2, and 2 ATP generated via substrate level phosphorylation. Okay, that is a lot of stuff going on. So your reactants here are 2 acetyl coenzymes A. That's it. And your products are 4 carbon dioxide, 6 NADH, 2 FADH, and 2 ATP via substrate level phosphorylation. I want to mention here with the carbon dioxide, it says there are 4 carbon dioxides generated. Notice that what's entering the Krebs cycle are 2 carbon molecules. The reason so that would be 2 carbon molecules would mean that 4 carbons total are entering the Krebs cycle. 4 carbons enter the Krebs cycle in the form of acetyl coenzyme A. 4 carbons exit the Krebs cycle in the form of carbon dioxide. They bound with oxygen and are being exported out of the body to be recycled. Okay. Again, this tells you that the Krebs cycle definitely requires oxygen. So at this point, we know that the only portion of cellular respiration that did not require oxygen was glycolysis. Glycolysis didn't release any carbons. All it did was split glucose in half. No carbons were released, so no oxygen was required to bind up carbons. However, in the intermediate stage, carbon was chopped off, generated carbon dioxide by binding with oxygen. Same with the Krebs cycle. We're breaking apart, releasing carbons, and therefore using oxygen to get it out of the body. So again, we enter the Krebs cycle with a 2 carbon acetyl coenzyme A. Every 2 carbon acetyl coenzyme A that enters generates 3 NADH, 1 FADH2, and 1 ATP via substrate level phosphorylation. Because we have 2 acetyl coenzyme A, you're having 2 of these reactions take place. So the overall energy products generated via this are going to be 2 ATP via substrate level phosphorylation, 6 NADH, 2 FADH2. How do I know that those ATP were generated via substrate level phosphorylation? Did we see an electron transport chain yet? We didn't. Those ATP were generated when carbon was broken off, right? So they're at the level of the substrate, it's substrate level phosphorylation. We've had plenty of oxidation reactions, right? The fact that we have so far in our energy tally 2 NADH from glycolysis, 2 NADH from the intermediate stage, 6 NADH, and 2 FADH2 from the Krebs cycle tells us lots of oxidation has taken place. But no oxidative phosphorylation has taken place yet because we haven't seen an electron transport chain. Lots of oxidation. We're ready for our electron transport chain, right? All right. So take note of our energy tally so far. So far, we've generated 4 ATP via substrate level phosphorylation, net 2 from glycolysis, and 2 during the Krebs cycle. No ATP was generated during the intermediate cycle, but glycolysis, intermediate, and Krebs gave us a lot of electrons. We have a lot of electron carriers. Notice that after the Krebs cycle, we have nothing left of our initial carbon compound. We have completely oxidized it. We've got rid of all the carbons, we've got rid of all the bonds. All we are left with are the electrons that were carried in those bonds. Now, what are we going to do with those electrons? You know what we're going to do? Let's do it. Well, we take them to an electron transport chain. Okay, so this is when it is important to remember the thing that I told you in the last lecture. And I'm actually going to go back to that slide to remind you that when it comes to an electron transport chain and a proton motive force, every one hydrogen pumped across a membrane equates to 1 ATP. All right, so 1 hydrogen pumped equals 1 ATP. Okay, so let's move back to our electron transport chain slide. What happens when we have all of that NADH and that FADH2 reach the electron transport chain is those will release electrons, but they actually bind to different places in the electron transport chain. So you can kind of see that here. This slide's a little bit messy, but you can see it. So the electron transport chain is a chain of transport pumps that basically are fueled by electrons, and they transport hydrogens across a membrane. So you can see here what you're looking at here is the brown is a membrane. It's a mitochondrial membrane if it's a eukaryote, and it has 3 pumps, hydrogen pumps, and then you can see that those are all pre to ATP synthase. So ATP synthase it's sort of like a hydraulic kind of thing, where you generate this giant proton motive force, a giant kind of concentration gradient of hydrogens. And every time one hydrogen flies through ATP synthase, that's enough energy to generate 1 ATP. So NADH and FADH2 bind to different pumps. So what happens is NADH, which remember every NADH is carrying 1 electron. When NADH comes to the electron transport chain, it has enough energy to pump 3 hydrogens across this membrane. So note NADH is not carrying 3 hydrogens, right? It is only carrying 1. But there are hydrogens kind of hanging out in the mitochondrial matrix. The mitochondria tends to be a little acidic, meaning it has a lot of hydrogen ions anyway. So what happens is NADH gets to the mitochondria, and it has the ability to pump 1, 2, 3 hydrogens across the mitochondrial membrane. So remember our rule that every hydrogen equals 1 to ATP. That means that if NADH gets to the electron transport chain and it can pump 1, 2, 3 hydrogens across, that means that every NADH that gets to the electron transport chain will be able to generate 3 ATP. 3 hydrogen pumped means 3 ATP. Okay. FADH2, when it gets to the electron transport chain, binds differently, but it can pump only 2 hydrogens across that membrane. So it pumps 1, 2 hydrogens. Every hydrogen equals 1 ATP. So FADH2, because it can pump 2 hydrogens across, will result in the production of 2 ATP. So this is important because this gets into our calculation of how much ATP is generated via cellular respiration. So throughout those 3 first processes, glycolysis, intermediate, and Krebs, right? You look at your energy tally. We got 4 ATP via substrate level phosphorylation, via reactions that took place during glycolysis and during the Krebs cycle. But primarily what we did is we broke bonds, released electrons, and we had NAD or FAD grab those electrons and carry them to the electron transport chain. So during glycolysis, we had 2 electrons released. Those electrons were carried by 2 NADH to the electron transport chain. We had 2 NADH, meaning that we could transport 1, 2, 3, 1, 2, 3. So 6 hydrogens across a membrane. 6 hydrogens across a membrane means that we're going to get 6 ATP. So 2 NADH during glycolysis will give us 6 ATP. The intermediate stage also gave us 2 NADH, which means we can get 1, 2, 3 from one of them, 1, 2, 3 from another. So the intermediate stage will end up giving us 6 more ATP via oxidative phosphorylation. And then during the Krebs cycle, we get 6 NADH. So that's like 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. Basically like 6 times 3, which is 18 ATP. But then you also have 2 FADH, which FADH gives us 1, 2 plus 1, 2, so 4. So that's 18 plus 4, which is 22 ATP via oxidative phosphorylation via the Krebs cycle, plus 6 plus 6. Okay, so that is like a lot of ATP via oxidative phosphorylation. So again, I mentioned earlier, why would you want to do substrate level or why would you want to do oxidative phosphorylation when it seems way easier to do substrate level phosphorylation? The answer is, and you can see from our energy tally, during glycolysis, we got 2 ATP. I mean, like 4 if you don't count the 2 we used, right? Every time you break a bond, you can kind of like usually get like 2 ATP, all right, via substrate level phosphorylation. Not a lot, it's some, not a lot. But every time you break a bond and you release 1 electron, if an NADH grabs that electron, that will result in 3 ATP. So during glycolysis, we broke glucose in half, and yeah, that was like, oh, here's some ATP. But because we released those electrons, and those electrons were used in the electron transport chain, that ended up giving us so much ATP. So oxidative phosphorylation is just so much more efficient because NAD and FAD can pump so many more hydrogens, and every 1 hydrogen results in 1 ATP. So again, like I mentioned, after we go through this process, right, you can see that when NADH pumps those hydrogens across, great, right? Then hydrogen is used to fuel the production of ATP. It's that sort of like releasing the Hoover Dam style energy that is fueling ATP synthase. But notice after that, we kind of just like have electrons hanging out and also have some like free hydrogens hanging out. And we don't really want those ions hanging out. And so what we do is we actually, again, you can see that red molecule right here. We use oxygen to bind up both the electrons and the hydrogens. And when we do that, we generate water. So oxygen is used by the electron transport chain in aerobic cellular respiration as what we call a final electron acceptor, basically to bind up any of the electrons we just used throughout this process. All right. So again, this is just showing you the calculation. Every NADH that comes into an electron transport chain is going to fuel the pumping of 3 hydrogens across a membrane, because every 1 hydrogen equals 1 ATP. If we get 3 hydrogens pumped across, that's going to give us 1, 2, 3 ATP for every 1 NADH. FADH2 fuels the pumping of 2 hydrogens across the membrane. Because every 1 hydrogen equals 1 ATP, FADH2 will give us 2 ATP. And this is just looking at that process. I included this because I wanted to just mention that the molecules that kind of help carry the electrons from pump to pump are called cytochromes. So if you hear like cytochrome C oxidase or any cytochrome, those are the molecules that help move the electrons from place to place. The other day, somebody asked about it wasn't ubiquitin, it was like, I can't remember, but it was basically CoQ or cytochrome Q. That also is involved in the electron transport chain. So kind of helping move those electrons from pump to pump to fuel the production, movement of hydrogens across that membrane. It's worth noting that cyanide actually inhibits cytochromes. So one of the reasons cyanide is so toxic is because it breaks down, it stops electron transport chains. Therefore, you can't make ATP, and therefore you can't perform metabolism. Scary. All right. So this is a summary of cellular respiration. big summary. So during glycolysis, what we did is we broke 6-carbon glucose into 2 3-carbon pyruvates. This used 2 ATP, so even though it produced 4 ATP, we say it resulted in a net total production of 2 ATP. But also because we broke 2 bonds, we released 2 electrons and therefore generated 2 NADH. We didn't require oxygen at any of these steps, so it's an anaerobic process. We then had the intermediate stage, which took those 2 3-carbon molecule pyruvates, added a coenzyme, broke a bond and generated acetyl coenzyme A. Because 1 carbon was broken off, we bound that carbon to an oxygen and generated carbon dioxide. This occurs for each of the pyruvates, and because we're breaking 1 bond in each of these, 1 electron is released from each of those bonds we break, and therefore we're producing a total of 2 NADH. This does require oxygen to bind up that carbon and generate carbon dioxide, so it is an aerobic or oxygen requiring process. Then we get to the citric acid cycle or the Krebs cycle. This occurs for every entering acetyl coenzyme A. It basically, as we talked about, is a giant cycle, but what you're doing is you're breaking the bond between those final 2 carbons, releasing all of those carbons. So 2 carbons enter in a 2-carbon compound. Those carbons are released individually, bound to oxygen, so this is an aerobic process, to generate carbon dioxide. So that's 2 carbon dioxides for every entering acetyl coenzyme A, which means 4 carbon dioxides total. You can see that in this picture. Per reaction, you're going to generate, or per acetyl-CoA, you're going to get 1 ATP via substrate level phosphorylation, 3 NADH, and 1 FADH2, which means that total, because this occurs for 2 acetyl coenzyme A's, you get 2 ATP via substrate level phosphorylation, 6 NADH, and 2 FADH2. Then what we need is something to utilize all of those electrons that were released via oxidation of glucose in the first 3 steps, glycolysis, intermediate, and the citric acid or Krebs cycle. And we use those electrons in the electron transport chain. This is when you have those NADH and FADH2 release the electrons, and those electrons are used to fuel pumps that pump hydrogen across a membrane in the mitochondria in the case of eukaryotes. These form a proton motive force or a proton gradient, which is producing a ton of potential energy. And every time a hydrogen flies through ATP synthase, this is enough energy for every 1 hydrogen to generate 1 ATP. And then finally, you have all those extra electrons combine with hydrogen and oxygen to form water. So oxygen is your final electron acceptor. This is just another summary of the process, I think, from your textbook, kind of looking at the location of each of these. I'm going to go through it, but I'm going to let you use it for reference. This is another summary of that process, what occurs in each of those reactions, but this is really what I want to talk about right now. So let's talk about how much energy is generated via this process. And when I'm talking about energy, I'm talking about how much ATP is generated via this process. So during glycolysis, glycolysis is our only time during cellular respiration that ATP is actually required. So we do have an initial investment of 2 ATP, but via glycolysis, remember, directly in glycolysis, you have 4 ATP generated via substrate level phosphorylation. So because we invested 2, we subtract 2 from our product of 4, and we say that we get a net gain of 2 ATP via substrate level phosphorylation in glycolysis. During glycolysis, we also break 2 bonds, release 2 electrons, and therefore generate 2 NADH. Because of the fact that every 1 NADH will pump 3 hydrogens across a membrane, and every 1 hydrogen equals 1 ATP, we can calculate that if we get 2 NADH, we're going to get 6 ATP. Again, because 1 NADH equals 3 ATP, so 3 times 2 is 6. Okay, so 6 ATP generated via oxidative phosphorylation. So glycolysis will give us 2 ATP net via substrate level phosphorylation, and will give us 6 ATP via oxidative phosphorylation. The intermediate stage doesn't require any ATP. We break a bond, one bond in each of the 2 pyruvates. Every bond gives us 1 electron. 1 electron means 1 NADH because there's 2 pyruvates. That means 2 bonds were broken, therefore 2 electrons were released, therefore 2 NADH. Remember, every 1 NADH equals 3 ATP, so 3 times 2, intermediate stage, gives us 6 ATP via oxidative phosphorylation. Then we have the Krebs cycle, which is crazy. First, it gives us 2 ATP via substrate level phosphorylation, just gives us some ATP. But then we're breaking a ton of bonds. And overall, with our 2 entering acetyl coenzyme molecules, we'll get output of 6 NADH and 2 FADH2s. Since every 1 NADH gives us 3 ATP, we can say 6 times 3 gives us 18 ATP. And because every 1 FADH2 gives us 2 ATP, we can say 2 times 2 gives us 4 ATP. So remember, because NADH and FADH require an electron transport chain to generate this ATP, that's oxidative phosphorylation. So total, if we add up all of our ATP, we had net 2 via substrate level phosphorylation and glycolysis, 6 via oxidative phosphorylation resulting from glycolysis, 6 via oxidative phosphorylation during intermediate stage, 2 via substrate level phosphorylation during Krebs cycle, 18 from oxidative phosphorylation during Krebs cycle from NADH, 4 from FADH2 oxidative phosphorylation resulting from the Krebs cycle. If we add all of that together, that gives us 38 total ATP. 1 6-carbon sugar. 1 6-carbon sugar gave us 38 ATP. If you count for the fact that every freaking huge reaction, cellular respiration, is a huge metabolic pathway, and it only required 2 ATP, you generated 38. That means every 1 time cellular respiration occurs, you can get 16 more cellular respiration reactions. It is so insanely efficient. Okay, that's why we care so much about cellular respiration. It is insane how much energy it can generate. So what I want you to be able to do with this information, I do want you to know that 38 ATP are generated via cellular respiration, and how. How many of these are generated via glycolysis, intermediate, and Krebs, and how? Was it substrate level phosphorylation or was it oxidative phosphorylation? You need to know. I could, on the exam or on the final, ask you how many ATP are generated via substrate level phosphorylation during the Krebs cycle. Your answer would need to be 2. I could ask you how many ATP are generated via oxidative phosphorylation in the intermediate stage. The answer would be 6, right? So you need to know how we got that ATP, both method, substrate level versus oxidative phosphorylation, and you need to be able to distinguish what is happening in each of these, both energetically and to your organic compound. So something I want to mention about cellular respiration is we tend to really focus, oh wait, that's not what I want to talk about. Okay, well, we tend to really focus on like glucose being our substrate for cellular respiration. So you can see here shown in blue, Carbohydrates, such as glucose, are a really good way of generating a ton of ATP, as we saw, because they can easily enter glycolysis, then go through intermediate, and then go through the Kreb cycle, and then the electron transport chain, and generate a ton of ATP. So it is very efficient. But that's not to say that carbohydrates, like sugars, like glucose, those are not the only organic molecule that can generate energy, obviously. We consume a lot more than just sugar, most of us at least. We consume also fats and proteins. And fats and proteins can also be utilized via pathways in cellular respiration to generate energy. In terms of fats, which are composed of a glycerol head and a fatty acid tail, the glycerol head of fats can actually enter glycolysis and generate ATP via substrate level phosphorylation, actually directly, and also oxidative phosphorylation. And the fatty acid tail, which are basically just giant hydrocarbons, actually can enter directly into the Krebs cycle and start generating a ton of ATP, primarily via oxidative phosphorylation. Proteins can be broken down into amino acids. The amino groups can be recycled for biosynthesis, but the carbon-containing compounds or the carbon-containing parts of amino acids, whether that's the R group or the carboxyl group, can actually enter into pyruvate oxidation, or it can enter directly into the Krebs cycle to also generate ATP. So the reason I mention this is because it's important to realize that not only sugar interact with cellular respiration, but you can have all macromolecules enter cellular respiration for utilization of production of energy at any given time. Right. I also want to mention that cellular respiration is not only important for generating energy, ATP, but cellular respiration is also extremely important for breaking down molecules like fats and proteins and carbohydrates and generating precursors for biosynthetic pathways. And remember, biosynthesis means the creation of macromolecules. So if you look at this, this is kind of looking at it backwards, but what you can see is that you have energy invested into any of these stages. So you have, this again is looking like backwards, but if you go through glycolysis through the Krebs cycle, byproducts of glycolysis can be utilized to create carbohydrates. Byproducts of glycolysis actually can go into the creation of fats and fatty acids. Byproducts of pyruvate and the Krebs cycle actually can go directly into the synthesis of proteins. So I want to mention that because cellular respiration is notable primarily, at least in textbooks, it's notable because it generates obviously a ton of ATP, but it is a central metabolic pathway not only for the production of ATP, but actually for the production of the vast majority of the macromolecules in your body. The vast majority of biosynthetic reactions, creation of fats and carbohydrates and amino acids in your body, requires cellular respiration or catabolism of ingested macromolecules to create precursors for your biosynthetic reactions as well. So it is a central metabolic pathway not only for catabolism and generation of ATP, but also for biosynthesis and generating precursors that are used in anabolic reactions to build your macromolecules. And this is just kind of comparing, because a lot of people confuse cellular respiration with respiration. They are linked, right? You do need oxygen for cellular respiration to bind up that carbon dioxide and get it out of the body. But those are two different things. Respiration is like kind of inhalation and exhalation. So breathing in oxygen, breathing out. But realistically, the purpose of respiration is to get that oxygen to your tissues so that your tissues can perform cellular respiration. And then breathing it in out is breathing out all that carbon that you broke down in the production of ATP. How cool is that? All right. I do want to compare something as well, a couple of things. So first off, we've talked a lot about the requirements of oxygen in cellular respiration. Cellular respiration requires oxygen to bind up released carbon and get it out of the body, but it also requires oxygen. Aerobic cellular respiration requires oxygen to be a final electron acceptor for the electron transport chain to get those electrons out of the mitochondria. Breathe it out, or sweat it out, I guess, because it's water. But your body also performs what's called anaerobic cellular respiration. And this means that your body can go through all of those 3 catabolic steps, right? Glycolysis, intermediate, and the Kreb cycle. And it can do the electron transport chain, and it can do that in the absence of oxygen. And it can do that if it just uses just a different electron acceptor. So just not oxygen as a final electron acceptor. You'll note that in this case, like oxygen is still required for intermediate and Kreb cycle, right? You still have CO2 released, but it is not required for the electron transport chain. And that's called anaerobic cellular respiration. So some examples of final electron acceptors that are not oxygen can be like sulfate or nitrite. Some others exist, but just note that in this case, if you're not using oxygen as a final electron acceptor, you're not generating water as a byproduct, right? As well as that, you can see that there's slight reduction in the amount of ATP produced. That is just because using a non-oxygen electron acceptor just requires a little bit of energy investment as well. So I want to point out something that's very important here. Aerobic cellular respiration and anaerobic cellular respiration are exactly the same for glycolysis, intermediate, and the Kreb cycle. The only thing that differs is the final electron acceptor after the electron transport chain. So everything else is the same. And I also want to mention that aside from going through the entire cellular respiration, sometimes your body doesn't have enough energy to kind of like send it through all of those stages. Or maybe you are going to not have a mitochondria present or something. And in that case, what you can do is what's called fermentation. Fermentation is just when you only undergo glycolysis. So what happens is you undergo glycolysis, you split glucose in half. You do remember via glycolysis, you generate a little bit of ATP via substrate level phosphorylation, but because with fermentation, you are breaking bonds, you're releasing electrons, but you don't have an electron transport chain to kind of use those electrons. A really important thing that happens during fermentation is that you somehow have to get rid of the electrons that are being carried by NADH and recycle that NADH into NAD+, so that the cycle can continue. So in order to do that, what you have as an electron acceptor is some sort of organic compound. So what happens is you have glycolysis take place. You have glucose split in two. You have electrons released, and you form NADH. But then that NADH releases the electron, and instead of like using that electron in an electron transport chain, that electron reduces an organic compound. So what happens is that organic compound tends to become acidic. So the product, well, or basic, the product of fermentation is some sort of reduced organic compound. Usually this will be some sort of like acids or alcohols, sometimes it's the gases, but the product of that varies kind of depending on what is being fermented. And again, this does not generate a lot of ATP. You can see ATP produced is 2. That's because glycolysis requires 2. It generates 4 ATP via substrate level phosphorylation, but because those 2 ATP were required, your only net gain is 2 ATP. So fermentation is not a very efficient energy generating pathway. This slide kind of goes through the process of fermentation. I'm not going to go through it because we kind of explained it on the previous slide. And this slide I love because it compares all 3 of these metabolic pathways. So we started out, we went through at the beginning of this lecture very much in depth the process of aerobic cellular respiration. I do want to remind you that aerobic cellular respiration takes place in 4 steps: glycolysis, intermediate stage, the Kreb cycle, the electron transport chain. The first 3 steps are when glucose is completely oxidized. You do get a little production of ATP via substrate level phosphorylation during glycolysis in the Kreb cycle, but primarily what you're doing is breaking bonds, releasing electrons, and binding up those electrons with electron carriers. And those electron carriers carry those electrons to an electron transport chain where a proton motive force is established to fuel the production of a ton of ATP. Then you have to get those electrons out of there, and the reason this is aerobic cellular respiration is because oxygen is used as a final electron acceptor. That's what gets those oxygens out of there, generating water as a byproduct. With anaerobic cellular respiration, note that this is exactly the same. Glycolysis, intermediate, and Krebs, a little bit of ATP, a lot of electrons released, therefore a lot of electron carriers, bringing these electrons to the electron transport chain. The only difference here is that instead of using oxygen as a final electron acceptor, you're using something else. Anything else? And then you can see that a huge difference is fermentation. Fermentation is only glycolysis. Glycolysis does not require oxygen because you're not releasing any carbons. So fermentation does not require oxygen. It is an anaerobic process. That doesn't mean it is anaerobic cellular respiration. Cellular respiration is glycolysis, intermediate, and Krebs, plus an electron transport chain. Fermentation is anaerobic because it doesn't require oxygen, but it is not cellular respiration. You only have glycolysis. You have a little bit of ATP produced via substrate level phosphorylation, net gain of 2 ATP compared to way more generated via cellular respiration. But because of the fact that you're using NAD, you have to recycle that into more NAD+. You kind of have to recycle that. So you end up having to reduce an organic compound in order to kind of get rid of the electron off NAD+ to make more NAD so the cycle can continue. And this ends up generating alcohol or acids or gases. You don't necessarily need to know the specifics of fermentation for this. All I want you to know is that it is literally only glycolysis. It doesn't require oxygen. And the final electron acceptor is an organic compound. So this could be lactic acid, which is shown here, which is a common one for our muscles. All right, so that is cellular respiration. I feel confident that you got it. So next time I talk to you, I'm going to talk to you about photosynthesis.